Welcome to the last part of the replication, that is telomere replication. In the previous videos we have been discussing about what is replication, what is a DNA replication, what is enzymes involved in it. First part was regarding the enzymes, second part what are the prokaryotic replication, next part was regarding the replication in eukaryote, and this part is the last part where we are going to discuss about the telomere replication as well as certain inhibitors which are involved in the process of replication. So let's begin. Before that, what is a telomere? So telomere are the physical end of the eukaryotic chromosome. So if you see, that is, this is a, if you see, this is a chromosome, okay, this is a chromosome, okay, this is a chromosome. The physical end of this chromosome is known as the telomere. Okay, what is the function of this telomere? It protects the chromosome from DNA degradation as well as from the end, that end point to get fused. For the end DNA end fusion and recombination, it protects the chromosome. Okay, the main function is its protection, and the telomere replication is semi-conservative in nature and it maintains the length of the chromosome. This is a very important point. What is the important function of the telomere? And it is regulate the cellular lifespan and is also considered as an agent that is responsible for aging. That is, after each replication of eukaryotic chromosome, the telomere gets shortened and as a result the aging process takes place. So telomere are the physical end of the eukaryotic chromosome. Second, they protect the chromosome from getting fused or degraded. Next, they have semi-conservative type of replication and they are responsible for the aging process and also maintain the length of the chromosome. Next, we will study about how the replication of the telomere takes place. Okay. Before that, let's understand why there is a problem in the telomere in the eukaryotic chromosome, because in the last part of the video I have told you that the eukaryotic replication does not end there. There is another part also, that is telomere replication. What happens in the telomere replication? What happens in the eukaryotic chromosome? So these are the two strands, okay, these are the two strands of the DNA. What happens? The 3' end of the chromosome, the 3' of the end of the DNA goes on replicating or goes on replicating, but the 5' end ends here. Okay, there is no more template left for the 5' region to get replication or to complete the replication, as a result of which this 3' gets overhang. To complete this part of the replication, the telomerase enzyme is helpful for to complete the replication part. Okay, what happens? Let's study. So in eukaryotes, replicating the ends of the lagging strand becomes a problem and there is no template available for the RNA primer of the last Okazaki fragment to bind. What happens? These are the two strands. After the remove this, here the primer will be attached to it, okay, because you know that the DNA moves from the RNA polymerase move from in this direction, 5' to 3' direction. So after the removal of the primer, this part remains blank. Okay, there is no more template available for this part of the plant to get completed. Okay, that's why this 3' end gets overhang. Okay, as a result, there is an overhang the telomere with a gradual shortening of the chromosome after each round of the replication. Okay, then what is the solution? The telomerase enzyme, an RNA protein enzyme that carries an RNA template and recognize the G-rich tip of the telomere sequence and extend it in the 5' to 3' direction before the replication. What happens? So here you can see this part is lagging, this part is empty. So the telomerase enzyme will come and bind here, and what it will do? It will help in the replication in the 5'. So it will provide an RNA primer. Okay, it will provide an RNA primer, and as a result of which a DNA polymerase will bind to it and complete the replication process, okay, in the 5' to 3' direction. From here to here, the 5 prime to 3 prime direction, the process will be completed. We will see in the diagram what it goes on. Okay. Consequently, the 3 prime end of the telomere is longer than the 5 prime end and forms at a loop which protect against the nucleus attack. Telomerase is not active in the somatic cell. This is very important, but it is active in the germ cell. Mostly they are seen in case of the stem cells. Okay. In the stem cells they are more active and they are species specific. According to species they vary. And they can only extend in the 3 prime OH end. From the 3 prime OH end only they can extend, and they use RNA as a template. You know what is a template, because in this first class I have told you template is a sample by which the sequence can be done, by the which the replication can be. It is a sample copy by seeing that the replication process can be exactly that replication process. Complementary primer binds to it and form a complementary strand, as a result of which the replication process can takes place. So how does this process is done? Let's see. So this is the parental strand. As I was telling you, this is the parental strand from 5 prime to 3 prime direction, and it is complete. Next is the 3 prime to 5 prime parental strand. See here, this strand 3 prime to 5 prime strand is incomplete, newly synthesized lagging strand. Okay, because it is half complete. Next, what happened? The telomerase enzyme will bind. Okay, here see, the telomerase enzyme will bind, and the telomerase enzyme moves from the what direction? I have told you, that is from the 5 prime to the 3 prime direction. Okay, it will extend from the 5 prime to 3 prime direction. You can see here from this side the direction of the telomerase synthesis. From this direction the telomerase synthesis will take place. Okay, so the telomere contains certain sequences. It is an RNA template. Telomerase with the bound RNA template. It will provide a primer that is made RNA. Okay, and to that the DNA polymerase will bind and complete the strand. So what happened? It contains certain sequences to which it is complementary to that of the parental strand. You can see A is complemented to T, G is complement to C. Okay. Telomerase will bound to the DNA. Then what happened? The telomerase extends in the 3 prime direction. What will happen? After binding to the telomerase enzyme will extend in this direction. It will extend the 3 prime side to in this direction. You can see here up to this the parental strand was present. Now this is the extra sequence which the telomerase enzyme will bind. Okay, this is the extra sequence which the telomerase enzyme has formed. Okay, after that what happened? After completion of lagging strand by the DNA polymerase, this will happen. So what happened? This is the extra strand. Now the DNA polymerase will bind here. Okay, the DNA polymerase will bind here, and finally what will happen? It will move in this direction. The DNA polymerase start extending in this direction. Okay, the DNA polymerase will complete this strand. You can see it is complementary to this strand and will complete the strand. So this is how the telomere replication takes place. Next is, certain inhibitors are present which mainly what they do? They provide certain inhibition effect in the process of replication. Like the bacterial gyrase is mainly inhibited by novobiosine and nalodexin. These are mainly asked in the exam. Okay, and mammalian topoisomerase is inhibited by toposid, and DNA breakage and rejoining process is mainly inhibited by ciprofloxacin, quinols and fluoroquinols. They will ask in the match the following, or they will ask in the multiple choice question. Okay, and DNA binding is mainly inhibited by actinomycin. Okay, and cross link between the complementary strand, there will be a cross link that will also be inhibited by the mitocyanin. So these are certain inhibitors, and this is how the telomerase that is telomere replication takes place. And this is about the end of the replication process, from starting how the replication took place, what are the enzymes involved, what are the how the prokaryotic, what is the eukaryotic, and how the telomere enzyme as telomerase replication as well as the inhibition is completed in this part of the video. With this we come to the end of the replication. In the next part of the video series we will be coming regarding certain other interesting topic. Hope you like this video. Do share among your friends, like and subscribe to my channel. Thank you and have a nice day.